Good evening, everybody, or good afternoon, everybody. My topic today is Beyond Distribution Modeling, Integrating Species Attributes to Predict Spatially Explicit Impact. And again, to thank Hans and Malulu to give me this chance to share you some our joint work about Ophraella communa and also its host plant Ambrosia artemisifolia. So from my talk today, I will just mainly to introduce you our one major tools for this talk, which is species distribution model. And I just use one slide to start because I like this sentence a lot that published on Annual Review of Ecology and Evolution Systematics on 2009. It says species distribution models are one step towards bringing science into policy making. So it makes people easier to understand what as a scientist, what our study actually are talking about. There are three components I'm gonna talk about today. One is just very general, everybody probably know it very well, the species distribution model. And then the second part is how we move it forward towards the better understanding or predicting the population level, which is population density prediction model. Then together, we want to use these tools to help us understanding better the species interaction spatially. So for a species distribution model, I think just very briefly to say, this is basically just by observation, the occurrences usually are present state. But if you have absence data would be great, but that's not very common situation. To correlate or statistically correlated with the environmental data could be anything that's very important for the life history of the target species. And that's help us to predict the geographic space. Meanwhile, with all the data we collected, we could also just estimate the current or the realistic niche based on the climatic data or any other environment data. And then this is the one example about Ambrosia artemisifolia that we did in 2017 with the open black circle or the occurrences we collected either on the from a lot of sources to show you the suitable area based on the model for the current climatic condition. And I just quickly Googled it, actually only for Ambrosia artemisifolia, this species, in the past several years, even this year, we still continuing have this species distribution model prediction for many reasons. Of course, to compare with other species or to estimate their distribution in different ranges or under different climatic conditions, present or futures, and so on. So it's very much widely used tools. This is another example that we currently do, not really ragweed, but the Bidens pilosa, another invasive species in China, like we also including other biotic factors like phylogenetic richness of the plant or the human influence and so on. But it's a little bit irrelevant, so actually I hide it. Okay, then come to today's, really we're interested is the biological control for Ambrosia. So we know that for current situation is if we have, we made this opinion paper last year to give a little bit idea when we should really consider biological control. At the end, we really propose that we should never ever exclude biological control management. So how to predict the efficacy of biological control management would be a very important question. So this comes to what Heinz said, the updated occurrences map of Ophraella communa that I assume everybody for today's talk must know this beetle very well. So the different color here represent the year that, thanks to everybody who sent me the occurrences, who contribute to this updated map. That's from 2013, the very first year when we observed it in the North Italy and the south of Switzerland. Up to today, this few months ago, people still find it either in France or I think Hungary and Serbia and so on. Then I overlay it with the suitable area of the beetle based on the model. But this suitability prediction is based on the data we collected in 2017. So apparently, currently, the suitability map should be very different because we have a lot of updated data. For example, just give one example. The one here in Romania.
Bucharest.
Yeah. You see, the suitable habitat is actually not very low, very high. It's actually even below the threshold, the optimal threshold, but it occurred. So based on the very limited data, we cannot make very good suitability prediction. That's why continuing collect the current occurrence data will also help us to make the prediction of the potential distribution of this beetle. And I guess everybody received the email from Malilu that attached with Excel sheet that if you could kindly send us back what you happen to find Ophraella in where you ever traveled, that would be great. And thank you very much for that. Okay, so what do we do then? Briefly, for the distribution, we could easily just overlay the distribution of the plant, which here indicate by the green, and the distribution of the beetle, which indicate by the red color. Then we know that it is the suitable area for both of these species. And that means this could be the potential biological control area. And in this way, we actually don't know the efficacy. We only know this beetle have higher chance to survive, to start to build the populations. But you can still see compared to the native range, we have really much less area of the overlap area in introduced European places, countries. So why that? So based on their realized niche, we could tell. So this is the animation, but I think it doesn't really matter to understand it. You can see here it's the solid line, very much the large solid line represent the total climatic environment condition that we use of the background, either in the North America or in the US. And the dashed line represent the most dominant climatic condition. So you see in this small area, that's this cloud indicate the high density of the occurrence we found. So it's not population density. It is occurrence, how often we found this occurrence of the ragweed. And you can see this very dark cloud area happen to be the very real environment condition in the North America. So it means it could be either this beetle either cannot really survive in this environment, or it could be it can survive there, but it just have no chance. So we need to really try that we really have to do experiment to examine this. So these are the currently so common used about a species distribution model method. And then come to the question, how to improve our prediction? How could we make better or the more quantitative evaluation of the biocontrol? So we think it's best is to include all the experimental mechanism that we could put in the models. For example, here, we try to really predict the abundance. So we correlate the field survey data of the pollen data and the density data of the ragweed. Then based on the every year annual density data, pollen data of the Ambrosia, we are able to estimate the current situation of Ambrosia population level across Europe. So this is a map, just to give you example, in 2019, the population density of Ambrosia, what happened in Europe. But of course, we can do that in Europe because we have a great pollen integrates data from all the pollen stations. And I have inquired in China. We have a lot of this air pollution control or monitoring stations, but they only monitor the grants, especially like the grants below PM 2.5, but they do not distinguish what species it is or even what type of the pollen it is. So then in China, this prediction, it will be very difficult. But in Europe, it's much more advanced, and you have this opportunity to really monitoring the Ambrosia density every year. Then for the beetle, you see this is a Ophraella communa. You see that for one life history, the one generation from mating to laying eggs, then 3 stages of the larvae and pupae, and again become adult. This takes about 25 days, depends on the temperature, of course. So temperature definitely impacts the developmental time. Another very crucial stage for the life history of the beetle is the humidity, because the egg batch cannot move. So you often see the egg batch are laying on the back side of the leaf, and this will limit the success rate of the hatching rate. Based on this the 2 key important factors that influence the population building up of the beetle, we then try to use this information to do either field monitoring and then lab work or experiment in the lab to calculate the population density in the spatial level. At the end, we hope to estimate the abundance of Ophraella. This is our first result. That's to predict the number of generations. So this is based on our colleague Benno Agostini's work that calculate the cumulative degree days. That means to make one generation from the first laying eggs until the next time when they become adults and lay eggs, it need how many degrees in Europe. And this would give us an idea. Then we calculate this based on the map, how much degree we actually have in Europe. We could estimate how many generations per year this beetle could make in specific location. So the darker green color here represent more generation numbers of this beetle. Then the second factor we said is the humidity. Humidity, we mimicked in total 6 different treatment, the humidity levels to calculate the hatching success of these beetles. And of course, based on the formula, we could estimate how much success they are making for the hatching success in each specific location or each cell. This is a 10 kilometer times 10 kilometer resolution. Now, we have these 2 very important factors. And, sorry. So for you to see the map a bit better. So this is we then create this formula based on temperature and relative humidity. We're able to make the prediction of the population increase of this beetle in Europe. Again, the darker color, the more population that actually increase. So you can see the highest population size of Ophraella is in either Portland or in coast of the Black Sea, Georgia, and so on. So this pretty much we can much in line with this observation that annual Ambrosia pollen reduction since 2013 by Ophraella about 80%. So these are this basically validate our prediction. Then based on this formula, we could also calculate the relative importance. So we know these are the 2 key environment factors. Then based on the change 1 degree of temperature or change 1 degree of relative humidity, we could calculate their relative importance. And we know now where or when, which situation temperature drives the population growth, and when relative humidity drives population growth. Again, we can transfer this formula back to the geographic map in Europe. So this will help us. So here, the red color means temperature drives the Ophraella population growth. Blue indicate red humidity drives population increase. So one day, if we really think about biological control release, the mass release strategy, I think this will give us a great first idea what the strategy we should do. If it's temperature driven, we probably should consider in the spring to release much more numbers to help us build up faster the population size. If it's driven by the relative humidity, so I think it's better to do more repetitive, repeat the release events. Now, besides the population prediction, this abundance prediction, we now also try to make the impact like ecological effects to count both climatic suitability and their impact magnitude for giving geographic area to help us to make the priority where we should do the first treatment. So both these models, these tools, all these fancy tools help us to link species distribution model with species traits and interactions, and help us building this population density models. And then hopefully at the end, we can do much better prediction of biological control efficacy. So this is one final example for Ambrosia that, again, based on the monitoring station of the pollen data and how much people or population that are sensitive to Ambrosia pollen, could estimate the number of grains per cubic meter of the air to estimate. Then this could help us to estimate how many people actually suffer from Ambrosia-induced allergies in Europe. And again, this can easily transfer to real money number. This will definitely make people easy to understand the impact by the Ambrosia admisifolia. And this is only the health cost, which is about 7.4 billion annually in Europe. So if we can do biocontrol by Ophraella in Europe, in the suitable area of the Ophraella, what we can do, or what we expected. We expected compared to before and after of the beetles release, we could estimate it's about 70% reduction of the patient in Europe, and it was saving the health cost about 1.1 billion euro per year. Now, we're back to our final conclusion is, again, SDMs are one step, or these models are one step towards bringing science into policy making. I hope I could at least convince you this is one tool we should often consider. Using spatial distribution model easily to have a geographically descriptions, and making much better distribution model in the population level, it will give us much better management strategies. Both together will help us predict species interaction much better and make the outcome of biocontrol prediction more precise. So this is the end of my talk, and thanks for a lot of people share the Ophraella data to us, and a lot of collaboratives, and also the collaboration with Heinz. Okay. Thank you.
Thank you very much for this very interesting presentation. As I know, the questions were sent to Ms. Yan in written form. If there are further questions in oral way, please take it.
We have a first question in the chat. So it was about the plant Bidens pilosa that you presented. So what is the harm of it for humans? Is it pollen, allergy, or other harm? And what is the problem for the ecosystems?
Okay, bidens is highly invasive in China. It's really everywhere, very common. And it is a very typical invasive plant that just reduces the biodiversity. Recently have visited one, we call that what, national park? Shenlongjia. I don't know if anybody know that. It's one of our biggest wetland, Dajiuhu area. It just covers completely by the bidens pilosa. In November when I visited there, it just and the seeds, it's saying like all this, you know, how to say that? This family, I forgot.
What?
This family.
Bidens?
Same as Ambrosia.
It's an asteraceous.
Yeah, asteraceous plants.
Composite.
It produces like dramatically amount of seeds, and this one attached to the animals, to humans, to the cars is very, very easily. It has these hairs on the seeds. So that's why we do that, because we got this. We have already previous publication, not by us but by other people. I see their prediction is much less than what I have visited the site. Then I got, okay, we probably can easily do this, and just guide a student, supervise a student to make such map. It's as I said, I just want to show this is a really not very difficult tools and widely used, and it's a really efficacy and relatively cheap.
What is a bit new here is this climatic and human influence. So also how human activities will contribute to the occurrences. As you have seen for Ophraella, it also occurs in Budapest, was first landing in Hungary, and Bucharest was the first landing in Romania. And this is often the case that in this airport area, it's a lot of ruderal land, and there also occurs ragweed. So if by air and freight or people, Ophraella is distributed, it can easily land there. So even if it's not the most suitable habitats, but they find the plant there. That's why often first occurrences are directly related to human activities. In this case here, trade or human transport.
Okay, I see another question is Ophraella and Ambrosia distribution do not coincide. So in Europe, if we introduce Ophraella to other terrestrials, what harm it can cause for other biota? That's a typical.
If we go back to the overlay ragweed.
Yeah, so this is a typical biocontrol safety risky and beneficial reason. I think this is to other biota. I don't think this is what the model can predict. The harm on other biota should be done by the experimentally examined. And I think for currently biocontrol project to do the very restrictive risk analysis and assessment experimentally. Not the model is only be able to tell you or help people to either identify the suitable area to find the species or where to release, how many to release, and the best, the priority of the release. It's more like before the experiment or after or before the release.
Yeah, it's a tricky thing. So see, you introduce something into the food chain. So you have ragweed, and then you have billions of this new biomass created by Ophraella, and you can assume that then other predators and parasitoids might find this new prey and build up high population. And then it's again these natural enemies of the natural enemy who could then also attack other insects at the level of the phytophagous insect. So there's like one tool I know is this network analysis, where you study the network in North America. So you see in North America how many phytophagous insects are on ragweed, how many parasitoids they have, and how many preys are shared by each of these predator and parasitoid. And then if you introduce that into the network in Europe, then you will probably see what that might cause. But see, this is like post-release monitoring is one of the big deficiencies in biocontrol programs. See, nobody up to now is looking at which predators and parasitoids jump on Ophraella. We don't know, but this would be a fantastic new study to do. Does that a bit reply to the question, I hope? Then the next one, where did you take data of the pollen for Ambrosia? This is based on this Nature Communication paper. Jen, can you still show how many pollen stations we had?
Yes.
So we had nearly 300 pollen monitoring stations across Europe, and we had 80 data sets from hospitals about sensitization rates. And then we had from several places specific sensitization rates only to Ambrosia artemisifolia. And then this helped to calculate overall sensitization rates across Europe only for ragweed. And I see Victoria, you know better. See, we checked very well that ragwort and Ambrosia, we can take them also phenologically apart, and also that really helped to be as precise as possible to come up with sensitization rates. And then we calculated nationally specific health costs, absences of work, medication, visits to doctors, and so on, to come up with these figures here of what Ambrosia is causing damage. And then when Ophraella would reach a suitable habitat niche, and as you can see, the next one, where the new map showed. No, the new map. See, you can see that. We have this data point in Hungary, but this is again the airport. But the latest experiments that are being done in the field, they show that the beetle could also survive east and north and south of Hungary in cages where we released ragweed. Okay, I hope this might also reply to this question. And then this one, Jen. You said something about genetics in the beginning. Can you tell me more? Because genetics of Ophraella communa is possibly part of my PhD thesis in the future. I already did genetics in Serbia, and I would like to continue with nearby countries. So, Jen, say something about genetics of Ophraella.
Okay. I'm not really sure. What do you mean, say much more? I guess it's like, do you have any specific area? If you are talking about draft genome, the draft genome, there's already a publication published on 2020, I believe, in May, in the GBE by Heinz group before.
Maybe Matija, you can ask in direct instead of writing if you want to.
Matija, yeah.
And that's
Hello?
the draft genome of Ophraella, if that's the question.
Yeah.
Just a second. Okay. Hello. For my master thesis, I did genetics of Ophraella communa in Serbia, and we discovered two populations in Serbia, And I just wanted, I'm going to continue possibly to do genetics on Ophraella communa for my PhD thesis in the future. So I would like to know more if you did anything with Ophraella in Europe for genetics, because one of my goals is to research genetics of Ophraella in nearby countries, maybe whole Europe. We'll see.
What do you mean you did some genetics in Serbia? What does that mean?
I did genetics with cytochrome oxidase subunit 1, last part of it, and discovered two populations existing in Serbia.
You mean there are two genotypes in Serbia?
That's right. That's right.
And you want to know the sources of the migration history of them, or? Yeah. Sources and I would like to know more about if you did any genetics in Europe with that about populations in Europe. Currently, there's nothing published, but we also try to look the migration history of the Ophraella in Europe and also in Asia, but nothing has been published yet.
See, we are presently working on a draft genome. In this paper that you can see here, we had very little DNA, so we had to pool an inbred population, only 2 males and 2 females, and we put them together. And then this made a horrible mess, because later there were also studies already done in China. And okay, they found out, they helped us out a bit to identify male and females, and we are continue working in it. So what you want to do is the whole network, because we collected specimen from Florida up to New York, and also left and right and south and north in China. And we collected in this distribution map from 2013, the first landing. We have samples, and now we have samples all over Europe. So we can also then identify which genotypes are spreading and things like that. So these studies are well underway also.
So no, we don't have genotypes of the population in France for the question of Marilou. No.
Okay, thank you.
No samples, sorry.
Yeah, it's Marilou. So I know that someone in the participants, Zoé Rousset, had master thesis on Ophraella communa in France. So I was asking her if her team did some genotype of it or not. But I don't know if you can hear me, Zoé.
So Zoe?
Yes. So we have the specimens that Marilou sent us, but I don't think any genetics have been done yet. But it's not in my team; it's another team in RAE. They have the specimen and they wanted to look at it, but not yet. Sorry.
Okay, thank you. Sorry, and Victoria want to ask a question too?
Yeah, I have some comments. For example, why I asked about bidens or bidens, because, for example, in here we have bidens tripartita. This is another species of the same genus, but it's considered to be the medicinal plant, for example, and you have it as invasive. And on the other hand, pollen usually is not distributed widely because this is an entomophilous plant. And another problem can be if people use it for, again, for making some teas or so. Those who have allergy for Asteratza can have the allergy also for bidens. But I was wondering if it is invasive, so you should know some exact harm for ecosystem. So probably you couldn't answer, but I did not here the clear answer what is the problem with these bidens, except that it is connecting with their clothes. This is common way of distribution of the species of this genus. So this is nothing wondering. This is like this. And another question, one more question is about the Ophraella in China. As far as I understand, you have some Ambrosia in China as well. And is it possible, for example, do you have Ophraella there? And is it possible, for example, to put it in, Ophraella in China into this loci of Ambrosia and see what will happen?
It is in China since 2000. And then in 2008, I think it has the first mass release experiments. So currently it's already being widely released in China, and they are naturally spreading by themselves. It's making quite sustainable populations, spreading mainly towards center and south of China. For the management efficiency, it's okay. But the point is, we have a lot of seed bank. So the dynamic, it's another thing we are continue doing is once Ophraella controlled the ragweed population, usually it's completely eliminated, but it's for that year. So you see, then Ophraella have no food and they cannot overwinter. Next year, Ophraella disappeared. But then ragweed start germinate from the seed bank, and they have to wait some Ophraella from other place to come here to do the control. So the management somehow, yes, it's very good because they build population pretty fast. But the point is there too much seed bank in the soil. And it's the dynamic, the ragweed become problem. Like 1 year, very low pollen, the next year, 2 years later, become a lot. Yeah, because the seeds continue coming out. And then this management is really not only spatially very important, also temporally very important. It's very different from year to year.
No, but still it is considered one of the biggest biocontrol success against an invasive species. It was also accidentally introduced, Ophraella. In 2001, yeah, it landed there. And then there were many host specificity tests, especially with regard to the risk of sunflower attack. And this was clarified and published 20 publications or more, then it was considered safe. After that point, we consider it as a biocontrol agent. Before it was like an accidental introduction. And then it was mass reared, as Jens said, 1 million Ophraella per greenhouse per month. This means one generation. And then they were released. And in so we did all the surveys in southern China towards Hong Kong area. The beetle has up to 5 generations easily. And if we go more north, they probably have up to 2 generations. So there's a lot of publications on Ophraella and its impact. There's another species, Epiblema, another moth, that has then deliberately been released against ragweed. So there's a lot of biocontrol activity in China going on. About bidens, it's a biodiversity species. It's competitive. It displaces other species. Not like ragweed. Okay. See, ragweed is not really a terrible competitor, but it is a weed in many crops because they cannot be killed by herbicides. Laszlo might probably say there's now a new herbicide that has been registered, I think this year for the first time also in Hungary, where they say you can take it out from sunflower fields. So let's see how far this is possible. And the other one is the human health impact. Okay, so for that point of view, ragweed is a bit special. All okay, like that?
Now I'm satisfied. Thank you.
Okay.
Further questions? If not, again.
Can I make can I just make a short communication?
Yes.
Yesterday, we tried to initiate a new working group within the EWRS, that's the European Weed Research Society. And yesterday was the General Assembly, and the new working group on biological control has been approved. We had a first meeting earlier this year in Aveiro in Portugal. That was the kickoff meeting. You surely have read about it. And now the new convener is Marion Sayer. She's a plant pathologist from CABI in UK. So this working group is now starting. So I'm very much looking forward to what's going on in the future with that. So we have Ophraella, we have many other cases, we have active groups in Portugal, we have submitted a cost action. So all this means that biocontrol will finally become a bit more prominent, hopefully also in Europe. Just as a quick information.
Thank you very much, Heinz. So if no more questions and comments, I thank you very much again for Ms. Jan for this great presentation. This is a great project with very valuable results. And probably we will not meet until the holidays. So I would like to wish you Merry Christmas and a Happy New Year. Our next talk on Ambrosia online session will be next year. You will be informed about it in time. Thank you very much for all of you.